Excellent. All right. Well, I'm so glad you're here. I have the nice chance to welcome Emma Bain to have a small career conversation with us and the students who are on the line. Emma, I have been very happy to get to know you a little bit and your work a little bit, and just thrilled that you can share your story with these students who will, I think, find it really inspiring. I think sometimes the best way to start is to ask what you're doing now, where you are, and what your day-to-day is like, and then maybe we'll back it up to the way you got there, if that's okay with you. I
Sure, definitely. Can everyone hear me okay?
can hear you great.
Okay, awesome. And I have a few slides too, so we can start kind of, as you said, with where I am now, and then maybe backtrack, and I can show those too if that's helpful.
Absolutely.
Great. So, as Natalie said, my name is Emma Bain. I am currently a fourth year neuroscience PhD candidate at Brandeis University. So, right now I'm Waltham at Brandeis. And so, as a PhD candidate, I am in the lab and conducting research for my thesis. And so, every day looks a bit different. Today, I was actually doing an experiment, which was exciting, and I was testing a different protein or small molecule that I purified. So, went through all the steps of isolating a special protein that I care about. And then I used a special type of microscope to look at single molecules of this protein that had a fluorescent label on it. So it was different colors, and I could actually see it come into view and watch its dynamics with other partners that it binds to or plays with in cells. This is an in vitro experiment because it is in a test tube, and I'm using just components of a living system, but separately. And so that's kind of related to my thesis work. But I've done a lot of different types of research while I've been here. That's kind of the beauty of graduate school, is that you can try a bunch of different things and see what sticks. I do have one project, and ultimately at the end of my PhD, I will defend, a cohesive story. But also, one of the large reasons I came to graduate school was to figure out what I wanted to do in the long term. And so, as you'll see as I go through my kind of career history, I've tried a bunch of different things before coming to graduate school, and sometimes that's unique because a lot of times students go right from college to graduate school if they know, they want to do that. I didn't know that I was going to do this. So at your age and stage, I could not have told you I would have ended up here. And that's kind of the fun of it too. You don't really know all the time, and you have to take it step by step, job by job to really figure out what you do like. Also, jobs, you learn what you dislike about things, and that's really helpful for kind of the long trajectory of your career because it will be a long time. So that's kind of what I'm working on now. But if you want me to back up, I'm happy to do that.
I would love that. Yeah, two quick things. One is
Yeah,
we're
great.
going to be doing an experiment about fluorescence in the next half an hour. So
Yes.
perfect timing that you're talking about fluorescence as your experimental work, because we're going to look at fluorescence with our hands-on experiment next. And then for sure, people have careers that sort of meander and start in one place and digress and then go back to something else. So I would love to hear that part of it. I have given you the power to share your slide if you would like to do that.
Perfect. So I'll start by doing that. And as I'm talking, feel free to interrupt or ask questions in the chat because I'm an open book and this is for you guys. So I will share my slides. I have a few, so we'll see what we get to. So you're
Great.
seeing that.
Yeah.
You see all of them, and let's make this bigger. Oops. Are you seeing this too? Security alert. Okay.
Yeah.
Is that okay?
That looks great.
Perfect. So I'll just move this to the side. Okay. So I thought it would be helpful to do a bit of a timeline. There's a lot going on here, so I'll just start kind of from the left side. So I grew up in a town not too far from here on the North Shore called Marblehead, about 40 minutes north of Boston. And so just like all of you, kind of took the normal classes, but really fell in love with biology or started to notice that I liked the biology class more than chemistry or art or anything else, which was interesting because at that point you're kind of just figuring it out. You're going to decide kind of more in depth later on. But that's what I noticed. Then I went to Middlebury College, which is a small liberal arts college in Middlebury, Vermont. And so if you guys know Harry Potter, it looks like Hogwarts. So that was not the only reason I went, but it was beautiful campus, definitely cold, but really, really beautiful. And there is where I kind of honed in specifically on what kind of science I liked, which tended to be neuroscience, which is the study of the brain and the nervous system. Kind of, there's a lot that goes into it. But specifically, I was just really fascinated by the brain and what we know about it. There's obviously a lot we don't know about it still, but I wanted to kind of learn more. And at that time, Middlebury hadn't had a full major for neuroscience, so it was more of a department or a collaborative program. So I was taking classes that were in biology, but also psychology and philosophy.
Hmm.
So I really got the holistic perspective of neuroscience, which is fantastic. But what I did do there that kind of set the tone for where I was to go next was I started doing research in a lab. And so the summer after I went abroad, I knew I wanted to stay on campus. And a lot of my friends were working or volunteering in various research labs. So I said, perfect. And I volunteered in one of those. And our lab specifically focused on mindfulness and meditation. So it was a very cool thing to study. And in the summer in particular, we went out and recruited participants in the nearby towns in Burlington, Vermont, the cities that were close by. And we invited those participants in and actually conducted a study where we kind of rigged a whole scenario where we had them do different assessments and tests, if you will, and kind of told them that there was another participant in the other room when there wasn't. But we wanted to test how competitive they would be or compassionate if this person was struggling or needed something, even though there was no person there. But then so we compared people who did meditate and considered themselves mindful to people who did not. And then at the end, we collected saliva samples. So actually wanted to see their hormone levels, so testosterone and cortisol, which is a stress hormone. But that part took place outside of our hands. But it was my first taste of conducting research in a clinical setting, so interacting with people. And we were lucky enough to present our research and findings once in California. So we actually went to Stanford my senior year to do that. And the photo on the bottom left is me and a colleague presenting our work. And the other one was right down the road at UVM. And it was there that I really determined that I really liked doing research, sharing research with other people, hearing what they're working on and new questions they're asking. And so that kind of told me, okay, research is definitely a path I want to pursue. What jobs are available that will get me towards research or involve research in some way? And so in my final months in college, I started applying to jobs. And I also, being from and around the Boston area, I knew there was a lot going on right here. So it was a perfect place to start looking. And one of the first jobs and job interviews was at Brigham and Women's Hospital in Boston as a technical research assistant.
Hmm.
So that felt perfect because research is in the title. And it was very nice because they were looking for people right out of college, minimal experience, which I definitely had at the time. And so I got the job there, which was a great experience. The top photo is me and a different colleague, and the Brigham and Women's Hospital kind of foyer is behind us. It was actually really pretty. And so it's in, right in the hospital was our lab. And my advisor or boss was an MD, PhD. So he saw patients as well as did research. So we were on the research side of his work. And it was actually really interesting work. It was in infertility and specifically endometriosis, which is a serious condition in women. So some of the things that I worked on was actually collecting samples or going up to the infertility clinic, getting samples, processing them, and then culturing cells with some of these samples to try to differentiate different cells and study the disease actually in a dish and in cell culture using stem cells. So that was super fascinating. Also, cell culture is a big skill and technique that you probably have heard a lot about. So it was great to kind of build my skill set, learn a ton about what it's like working in a real working lab in an academic hospital. But ultimately, while I enjoyed that, about a year and a half in, decided, okay, this is great, but definitely not what I want to do long term. So I started to kind of look at other avenues. And actually, the second conversation that would lead to my second job happened pretty serendipitously. I was with my family, and there was a family friend at the table next to us, and we got to talking. And he worked at Sanofi Genzyme, which is now, I think, Sanofi, but previously Genzyme out of Framingham. And he said there was an opening in neuroscience. And so I wasn't super seriously thinking of leaving until, that opportunity came up, and it seemed like the perfect opportunity and thing to try. And actually, the person I met with first was Lisa Stanek, who you guys also heard from. So it's very full circle. But I interviewed with her and the team over at Sanofi, and specifically in the genetic neurologic disorders and diseases area. And I really loved kind of everyone I met, and luckily I got the job. And so I was there for almost 4 years as well. So I was moving around. I actually have my next slide is to do with that job because I thought it was pretty interesting and you guys might want to see. So as I said, I worked my way up, started as a research assistant, then became an associate. And I think ultimately by the time I left it, the title was scientist, which is very cool. But what I did there was work with different mouse models, specifically looking at two different diseases, Angelman syndrome, which is a neurodevelopmental disorder of the nervous system, which involves kind of cognitive impairment and various executive functioning problems. And then ALS, which you guys may have heard of. It's a neurodegenerative disorder of the motor neurons. So my job was mainly in characterizing the different mice models to see how similar are these mice to the disease, and if we have possible treatments or therapies, whether it was a drug in the food that they ate or an injection like a type of medicine, did we see enough of a kind of repair going on that maybe this drug therapy would be worth pursuing kind of, in the next stages or for human and clinical studies. So that was kind of the ultimate goal, was to see if we could identify anything that would help real people and translate what we were doing to humans. So some of these apparatus, I guess, or activities I did were the rotarod. So in the blue is kind of basically a mouse treadmill. So that's a test of mobility. We put the mice on this rod that rotates, and we measure the latency to fall. So if they're, having trouble moving or with coordination, they'll fall faster than normal mice. So that was one of the things I did a lot, which is funny because you have to be pretty quick to catch all the mice, and so they don't run away. You can kind of see them trying to leave. So that was interesting to figure out. The second thing, so in the orange, is called the T maze. So you put a mouse kind of in that first horizontal tube and have a kind of block at the end and have it choose with a reward, either the right or the left. And that is a test of memory or spatial learning. So mice that are impaired in that will not really care which direction the reward is at, and you'll be able to compare between that and controls. Over on the green is called the marble burying assay. This one's very nice because typically our normal mice, when they encounter a novel object in the wild, they will be really interested and tend to bury it. So you can see, and I guess the panel C, most of the marbles are gone because the mice that have been in this cage bury them. However, mice that aren't interested or more anxious may leave them alone. And so you can compare different patterns using this assay. And then the last one on the bottom is a test of social interactions. So you actually put a different neighbor mouse in this little apparatus and have your mouse sniff around. And if they have no interest, usually they will be interested in something, a mate or something like that. But mice who have Angelman syndrome do not care about, social cues or anything like that. So they'll mainly leave it alone. So this was a really cool way and firsthand to, work with the mice models and do different types of characterization of the diseases, comparing different therapies.
It
So
is
that was
so
great.
cool. It is. It's so interesting to think about the assays that you use with animal models, because the
Yeah.
students are the students here have done some assays, but from, bacteria to understand how the bacteria are doing what they do, or if they're doing what they do. And they are very different than these kinds of behavioral kinds of assays. But that word assay is used throughout, right? It's just used to say, what's happening. It's a test of something that is happening. So I think that's
Exactly.
really, it's such a different way of assaying the behavior and the biology of these systems. Yeah.
Yes, there's so many different ways to test what's
So
going on.
many. So
Yep.
many. Yep. Yeah. So I think it's wonderful. So this was your work at Sanofi, yeah?
Yes. Yes.
And then tell me just briefly about going to graduate school and your decision to go to graduate school. And then maybe, if you knew something back when you were making all these decisions that you wish you'd known, you could share that too.
Yes, no, absolutely. So great question. I think so. As you can see, I really liked my job at Sanofi. But as I was going on and kind of as the years went on, I was beginning to see um, where my future was going or what it would look like um, if I stayed where I was. So most of the people I was interacting with had their PhDs. But as I said, in high school, would have had no clue that that's what I would want to do until being in those roles where you see it all around you. So as I was going about it, I think something clicked or something happened. Here, I'll just have a quick sip. Where just talking to my different colleagues, I think I made the decision of, I think grad school, if for my future career, and if I want to open more doors down the line, maybe have my own group, I would need the higher degree. So that was really my decision to go back because I loved my job and I'm still thinking about the return and what that could look like. Um, excuse me.
It's okay.
Um, so I think it was just, the moment of seeing or thinking about my future and how to open more doors. So I applied and actually interviewed with Brandeis in January 2020.
Oh, gosh.
So
Wow.
as so, oh my gosh, losing the voice. But anyway, that brought me here, and fortunately, I was able to start grad school despite the pandemic and what was happening. Oh no,
It always happens when you're busy talking. I know.
I know.
Yeah.
Let's see. So anyway, that's what brought me here. I'm in my fourth year now and have done a lot of great things. Took some classes, TA'd a little bit, so teaching assistant, and actually got to teach my own course. So I do have one slide on that. And I just completed teaching this undergraduate course with a fellow grad student. And the whole course was about different microscopes. So I'm happy to answer any questions about that, especially if you guys are going to be working on fluorescence today. But the main thing about being in grad school, as I kind of started with, was that I really wanted to see every possibility and career path available now as I think about graduating in the next few years, knowing what industry is like and having had that job at Sanofi, which I was super happy at. But what else could be out there? So I tried my hand at teaching, which I liked a lot, and hopefully I can do more of. So I'm kind of trying it all out, which I think that would be my biggest advice is try everything.
Yeah, yeah. Excellent. I won't keep making you talk and drink. I
No, I'll
think
keep.
that that's a really great piece of advice is to, you know, not close off avenues, be open to those chance conversations with people at a table that might really like change what you're thinking and change your path, right? So,
Exactly.
yeah, I think that's awesome. And so, you're in your fourth year.
Yes.
And what does it look like for the next year, next 2 years for you?
So I'll be kind of forging ahead with my research project.
Mhm.
And that actually leads me to, I have one more slide.
Okay, yeah. Oh, you poor thing.
No, I don't.
I'm gonna try to make you stop so that you don't have to push your.
I'm just going to leave this up here. So
Yes.
part of the things we do in graduate school is publish papers. So we work really hard to come up with a story in the lab based on a specific project. So I was really lucky to have published this paper in March. And so one of the things before you graduate is try to publish a paper. So I have mine now, but I'm still
Okay.
working on some other stories and other projects.
Yeah.
And this is live if you guys are interested.
Absolutely. Well, congratulations, publishing a paper. And I see that you're the first author there. It is important to, you know, have first author papers that signals to the world that you did the bulk of the work there, although that may be that you and the second author shared co-authorship. But it's really, it's a wonderful and very high bar to be able to publish paper. So congratulations.
Thank you.
That is
And so,
awesome.
yeah.
Yeah, yeah. I don't know if the students have particular questions either about neuro or about being a PhD student at Brandeis, or if there are additional questions about, you know, career path changes. Oh, Ruby's asking what the favorite part of your work is. It's a great question, Ruby.
Great question. And I was also going to say, I'll leave my email. So if you guys come up with anything,
Great.
also
Thank you.
for, I don't have to speak for
Yeah.
responding to those. But I love that question. So I would say, so it's funny because I spoke a bunch about mice, but I actually am working with a different animal. Oh no,
Oh.
animal model in flies now.
Mm.
So the fruit fly, which I'm loving because they're a lot
Wow.
faster,
Yes.
really small, so I don't have to worry about handling them as much. But it's really, really fun too. So I
Yes.
would say that's
Yes.
my favorite
The
part.
fruit flies will not bite you, but they
Nope,
will fly away. You do have to chase them sometimes.
exactly,
Yeah.
exactly.
Yeah. Excellent. Awesome. Well, that is a really great question. So, yeah, fruit flies are amazing for genetic studies and things like
Yes,
that. So
yes.
awesome. That's really excellent. Do any of the other students have a question for Emma? She's wonderful. She's clearly super. Oh, thank you for your email. You know, clearly a great teacher. Running your own class as a graduate student is not common, not frequently done. Having a first author publication by your fourth year is also really great. So congratulations for that. Yeah, it sounds like you're thriving, and it's wonderful to see that, you know, there's not a direct path. Sometimes people feel like they have to just keep going from high school to college to graduate school to postdoc to job. And it's really a wonderful story to see that you've sort of navigated through your interests and through different paths and opportunities. So, and all here in Boston, right?
Yeah, all local.
Yeah.
So it can be done right here.
Yes. Yes. So for those who do not want to have to leave, there's lots and lots to do here. Excellent. Well, thank you, Emma, for your email and for your
Yes.
wonderful talk. It's really, like I say, it's great to have your work be about fluorescence. It's great to talk about assays. These are things that the students are focused on for the last week and a half of this program. And yeah, and teaching and next steps. And
Yeah, it's great they're learning this now. They're going to be set for the future.
They will be set. Yes, we want to make them ready for the future. That is our goal. So, awesome. Thank you so much. I really appreciate you being here. You're welcome to stick around. You do not have to.
All right. Thanks so much, guys.
Thanks. Bye.